Hello guys, welcome to Gene Allen School, and here we learn about the basic of genetic engineering and biotechnology. So guys, after a long time I have come, and today I will teach you a basic of genetic engineering, that is mRNA splicing type. Not the mRNA splicing, that is the one type of mRNA splicing, that is alternative splicing. mRNA splicing mechanism. So what is the basic of splicing? I'll show you what is the basic of splicing. Basic is that, suppose that is the mRNA and that is the intron and that are exon, that the colorful regions are exon region. This means the coding region. We know that exon are attached and make protein. So that is the intron side. We know that by splicing mechanism, the introns are spliced out from the mRNA. So here you see that when introns are spliced out by splicing mechanism, introns are out and exons are added to each other. Here is another type. Introns are out and exons are attached to each other. But there is a difference. You say that there is a yellow color and there is a green color. So you can ask me where there is a yellow color or there is a green color. We know that when we keep this yellow color exon and attach with the two exon and splice out the exon of green color, then a type of protein found. And when the yellow color of exon are spliced out with the introns and we get this type of exon and we found this protein. That is the mechanism of alternative splicing. But I will now teach you what is the splicing. Splicing is the splicing out of introns. That is the main fact. So the main fact of introns is spliced out of introns from mRNA and getting the protein from this mRNA. So these both type of splicing mechanism are splicing, but that is alternative splicing, because there is a difference from this to this protein. So I will now tell you about the alternative splicing. Suppose that is the mRNA. Normally we get a protein from this, but if we consider some differences among the exon region of this mRNA, we will get different protein. So the key point is that we will get different type of protein from an mRNA. That is called the splicing. So now you can easily understand what is the two type of proteins. So there is the mRNA, but from mRNA we are getting here two type of protein by splicing out the exon region. We are splicing out the introns. Normally what happens in the splicing mechanism, but we are creating different type of protein by creating different environment by splicing out the exon region of the mRNA. The alternative RNA splicing helps us to produce different variety of protein from a same single gene. By this type, we can get different type of protein that may be dangerous. Sometimes the proteins are dangerous, and sometimes the proteins are more valuable, and sometimes with better quality we found. So now we will learn about the alternative splicing. There is a mechanism figure, but here is the theoretical of alternative splicing. We know that particular exons of a gene may be included within or excluded from the final processed messenger RNA produced from the gene. Here they said that exons are included or exons are attached or exons are spliced out by alternative splicing. And here I say the different varieties of protein from a same gene. I said before that we will get different type of protein from a single gene. and that is very common. Alternate splicing very common in human body or eukaryotes. But that is very normal, because you know that in human genome 92% time happens. In human genome 92% times occurs in RNA transcription mechanism, this type. And let's say that, and at least 70% of the approximate 25,000 genes in the human genome undergo alternate splicing. So 25,000 genes of human genome undergo alternate splicing. So that is very common in our body. And now here we will learn how alternatively exons are attached with each other. You know that exons are attached and exons are spliced out, and some exons are skippable, and there is a differential method occurs among the genes, but there is some type of this alternative system. Here we will learn about the types. Here see that there is a skipped exon. That means one exon will skip out. One exon will not perform. That is skipped exon. So if you think that is an mRNA, we know that introns are not needed for our, but if this red, green and red, here is the three color exons are found, but here you see that by splicing mechanism two types of proteins are found. You can skip out this green exon and we will get two red exon. Here is the two red exon, and if you consider three exon, so we will get this protein. So from this mRNA we are getting two type of protein. So I hope you have understand about the skipped exon. The exons are skipped out, that will not perform. Here is it, and there is alternative 5' splice out. So in the alternative 5' here, the 5' splice site, there is a 5' splice and there is a 3' splice site. So here is something happens among this mRNA, that the 5' alternate splice site will attach with the exon. Here is the 5' splice site and alternative 3' splice site. So alternative 3' splice site, the 3' splice site will attach with the exon and create a new type of protein. And if you spliced out the 3' spliced, and you will get three type of exon and find this type of protein. Like this type of protein and this type of protein are same, because here is the same exon, but there is a difference among this protein and this protein, because here is a 5' splice site attached and here is a 3' splice site attached. And retained intron. Introns are retained maybe as sequence maybe by the figure you see that normally 3 exons are attached. But if you consider an intron, that is one type of skippable type. Introns are skipped out, but finally we will retain this exon. So retain this intron to this with the exon. But that is very rare in mammalians or then eukaryotes. That is very rare. And here you see that mutually exclusive exon. So exon will attach with each other mutually. That will attach with each, and that will attach with it. And this exon will not attach with this, and that will skip this, and the green will attach with the red. So these two types of mRNA or these two types of protein, we know that from this mRNA we will get different type of protein. So multiple exclusive exons. That is very simple, that they will attach with each other by their choice. It can attach with it, and then it will not attach with the green, but this two red color will attach with this color. So by and the next period you see that the green attach with red, and this green attach with red. So this type of protein is found. So these two type of proteins are found by the mutually exclusive exon. That By alternative splicing, we get different type of protein from a single mRNA, and that is very important for the variation among the protein sequence in our body, and we will get different type of protein from a mRNA. That is all about, and that is the importance in variation in our tissue, in our characters, in our body function, etc, etc. So I hope that everybody easily can now tell about alternative splicing. Thanks everyone. Subscribe my channel if you like my videos, and if you think that that is very easy for you to understand, share it to your friends. Thanks all. Goodbye.